Osmosis and osmotic pressure. Osmosis. It is defined as a movement of solvent molecules through a semi-permeable membrane from a less concentrated solution to a more concentrated solution. The must condition for this is the presence of semi-permeable membrane, which allows the passage of solvent molecules but blocks the passage of solute molecules. Osmosis can only take place when the two solutions with different concentrations of the same solute are placed in contact and are separated by a semi-permeable membrane. It will occur from the dilute solution into the concentrated solution or from the solution with the less solute concentration into the solution with higher solute concentration of the solute. Osmosis is a Greek word actually. In Greek, osmos means to push, and this was observed for the first time by Abbe Nollat. So in this picture we can see the solvent molecules on the solution side have a lower concentration than the molecules on the pure solvent side, and therefore the water molecules or the solvent molecules from the pure solvent side will move to the solution side. Before going ahead, we should also discuss about semi-permeable membranes. Actually, these are the membranes which allow the movement of the solvent molecules. The membranes appear to be continuous sheets or films, but they have very tiny sieves or pores which are semi-micro in nature. Through these sieves, only the molecules of the solvent can pass, while those of the solute, which are of bigger size, are not in a position to pass through. Semi-permeable membranes are of two types: natural, example is in that of the parchment paper or a bladder of goat or pig and the cells of plants, whereas artificial semi-permeable membranes include the semi-permeable membranes which are prepared chemically. For example, when aqueous solution of potassium ferrocyanide and copper sulfate are mixed in porous pot, the gelatinous precipitate of copper ferrocyanide acts as a semi-permeable membrane. So, osmotic pressure. In order to understand osmotic pressure, we have to see this like diagram. In this case, we have deposited the artificial semi-permeable membrane. For example, if we take that of the copper ferrocyanide over here in this U-shaped tube with the help of rubber stopper. Now we have to fill this with aqueous sugar solution in one side and water on the other side. Osmosis will occur and the level of the solution will slowly rise on the solution side. After a few days, the level will become stationary and no further osmosis will occur because hydrostatic pressure, called as osmotic pressure, develops on the surface of the solution which stops osmosis. So the osmotic pressure is defined as the excess hydrostatic pressure on the solution which prevent the movement of solvent molecules through semi-permeable membrane. The expression for this is given from the Boyle-Van 't Hoff law and Gay-Lussac's Van 't Hoff law, according to which pi is proportional to Ct, therefore pi equals to RCt, where pi is the osmotic pressure of the dilute solution. Isotonic solutions. Now, according to Van 't Hoff equation of the two dilute solutions, pi equals to CRT. This means that the two dilute solutions with same molar concentration at a given temperature have also same osmotic pressures. Such solutions are known as isotonic or iso-osmotic solutions. In case such solutions are placed in containers separated by semi-permeable membrane, no osmosis will occur. However, if the two solutions have different molar concentrations, they will have different osmotic pressures, and the solution having higher solute concentration is called hypertonic, while that with lesser concentration of the solute is known as hypotonic. Biological importance of osmosis is in the blood cells. If the solute concentration outside the cell is greater than that of the inside of the cell, the solution is hypertonic and water will flow out of the cell and crenation occurs. Also, if the solute concentration outside the cell is less than that of inside of the cell, the solution is hypotonic and hence water will flow into the cell and hemolysis results. What is reverse osmosis? If the pressure higher than the osmotic pressure is applied on the solution side, then the solvent will flow from the solution into the pure solvent through the semi-permeable membrane. This is called as reverse osmosis or RO. Its application resides in desalination of seawater, where seawater contains a large number of dissolved salts in it which is not fit for drinking. Seawater can be converted into potable water fit for drinking purpose by reverse osmosis process known as desalination. The schematic setup is shown over here, where pressure higher than the osmotic pressure is applied on seawater, which in fact is a solution. It is quite high and a variety of polymer membranes are available for this purpose. Basically, cellulose acetate membrane is quite useful. It is permeable to water but impermeable to the impurities present in seawater. Reverse osmosis has become very popular for the purification of drinking water, particularly in households as well. It is usually referred to as RO system. It has more or less become necessity, particularly in countries like India, where waterborne diseases are very common. Now, illustrative example on this. A solution containing 8.6 gram per liter of urea was found to be isotonic with a 5% solution of organic non-volatile solute. What is the molar mass of the organic solute? So we have to take it to be pi 1 and osmotic pressure of unknown compound as pi 2. Because pi 1 equals to pi 2, therefore CRT on both the sides, we will get the molecular mass of the organic solute as 348.9. There is one other example. What is the relationship between osmotic pressure of 10 grams of glucose and 10 grams of sucrose which are dissolved in 250 ml of water? So basically, we have to calculate the moles of glucose, urea and sucrose. More is the number of solute, higher will be the osmotic pressure. So N2 will be greater than that of N1, then N3. Therefore, pressure of urea will be highest, then that of glucose and then that of sucrose. Try solving this question yourself, where a dilute aqueous solution of non-dissociating compound containing 1.19 gram of the compound per liter of solution having osmotic pressure of 0.0288 atmosphere at a temperature of 37 degrees Celsius. Compute the molar mass of the compound.